Okay, good evening to all of you. And I guess it's 5:02, so we can start. And let me ask a question first. How many of you tried the activity um, after the last week? And have you guys came up with a solution to all the issues we faced? And if you can use the chat box and type to let me know the progress, whether you took a couple of steps forward, or you're still stuck, or whatever the situation you're in right now. So because we are going to repeat the whole process again, because you all need to be very familiar with the this whole procedure. So, and it will help you a lot. Okay, so just use the chat box and let me know uh, about your progress. There's no progress, so a silence means no progress. It's really hot here. I'm trying to swing the fan towards me. Give me 1 second. Okay, go. So now it's good. Okay, so I guess the silence is golden, right? So anyway, we will start and move forward with the activity. So if you can remember, the prerequisites are very similar to last time. You should have both files, and I kind of send you the instructions. Probably I think it's was yesterday, and also you need Galaxy accounts on both servers as far as for the today's requirement. But we are trying to focus on galaxy.eu, because even last time you experienced the issue, and also there's a small glitch with the fastp program in the galaxy, use galaxy.org, because it won't show us the output. It will be like a jumble. You need to download the file and then basically open it. So therefore I decided to go with the galaxy.eu, so that way hopefully the ride will be smooth. Okay, and if you can remember, just to give you a refresher, we were talking about a genome sequencing project focusing on identifying MRSA strains from patients. And so what we are going to do, like the last time, if not already, so please go ahead and change your Galaxy history name to um, from default to the new name, MRSA, MRSA. So go ahead and do it if not already. So we're going to move a little fast because these steps we already completed. And so when you're done changing the history, because I'm pretty sure you're very familiar with this, because if I can remember last time one of you had an issue, but I think we got it resolved. And make sure you are logged into the system, otherwise it won't let you rename the history. And also the earlier Galaxy versions, you need to click on the name unnamed history, so then it will expand and you can change it. But the latest versions, so you have this pencil icon and then you can click and move forward. And like I mentioned to you last time, so there will be a couple of Zoom questions. And so once you answer the question, so you will get some extra marks that can be used towards your final grade at the end. Okay. And first we are going to rename the files. But before that, I want to show you something. So let me quickly change the screen. Okay, so I ran the whole process on both .org and .eu. So therefore, for a fact, I know it's working. So if you are not yet uploaded your file, so you go to the file And then this time we are going to do a small change. Okay, so please listen very carefully. So we are going to make a collection because FastP is not going to recognize individual files. That was the main issue we experienced, because if you run the FastP using the command prompt, so this issue won't arises. But like, if you run it in the Galaxy environment, so you have only two options if you can remember. So you have the pair, or the like a single end versus the paired, but we need to upload it as a pair or paired collection. Okay, so I will show you how to make a collection. So you go to the collection, and then it should show you the two files you already uploaded. Okay, so go ahead and check your the Galaxy environment and see whether you can see both files. Okay, so if you can see both files, so please say yes. If you're facing any issues, you can either use chat box or unmute yourself and let me know. Meanwhile, I will show you. So maybe I can upload the file as well. So let me, okay. Okay, so I think by mean yes, you see both files, right? So what you need to do is, so let me grab my files and then drop them there first. Okay. So you basically you can drag and drop. Sometimes it won't work. So because last time it didn't work when I dropped files, yeah, it won't work. That's fine. You can go to the choose local files and then select to EC2 files. So you can see you can create a collection. One of the most important things you need to do here is basically from here. So under the options, you have the list under collection. So click on that and then select pair. Okay. And then for the type, because this one is B-shaped, so sometimes the program it takes some time to identify it. Couldn't see the Andika. What do you mean by couldn't see the field? No, we mean the files.
Yes, sir.
Okay, yeah. So what you can do is there are two ways to do it, but like easiest route you can take is to basically like what I did. So go ahead and like grab two files again. I hope you are using the galaxy.eu environment.
Yes, sir, I'm doing it.
Yeah. So if you can you like shall we go fresh? So that way it's easy. So upload like the two files like what I'm doing here. I will show you there's a workaround, but like so I think so otherwise uh, some might get confused with the whole process. Okay. So and then here under the type, so our one is fast Q, and then so here it's fast Q Sanger BC2. Okay, so please double check because there's another option called fast QC Sanger BC2. It is a very old format. Don't pick that, but just pick the fastq sanger.bc2. Okay. So like that. Okay, so you don't need to pick any reference. Okay, so then you just need to click start. Okay. Any questions? Any issues?
So if it's already uploaded, what can I?
Yeah, so there's a workaround. I'll show you in a bit. Give me 1 second. So I'm keeping this window open for like, maybe like another 30 seconds so others can see that we are using the pair and then the FastQC BCT, BC2, FastQ Sanger BC2 format. Okay. So, and I will upload it and then show you how to do it. Okay. So, and I can close it. So here you can see get data. So that. So, so collection operations. Okay, so there you can see build a list. So go ahead and you can click on that. And then it will give you options. Okay. So there are various options you can use. There's one with rules. So yes, so mine is uploading. Give me 1 second. Okay, I can change the environment and show you. I think I should have. Okay, so, okay, because I already have a collection. So can you basically click on the apply roles, and so let me go back to my latest environment. Okay, so here, can you see an option? The input collection. Just double check. Yes, no? Okay, so I'll show you another option. Okay, so no problem. So you click here, all right? And then select both files. And then if you look here, so this is the easiest way to go around. Go around the thing. So here you have option called build list of dataset pairs. Okay, you click on that. You can see. So it's going to combine one, the forward and reverse one. And here you can basically give a name. Okay, let me give an EGADF. Okay, and then I can click create a list of pairs. Is it clear?
Excuse me, sir.
Yep.
Sir, if I already uploaded my file as local file, it's not a collection,
That's fine. Yeah.
and it's appear on collection also. So I can choose the fast Q Sanger B Z, and the build option create the.
Yeah. So there are 3 ways to do it. So the data collection, yes, of course you can. So there you select pair, and then you select FASTQ Sanger .bc2. That's one option, and then you click build, and then the second one, this is the easiest one. What you can do is basically, so you click here the select button and select both, and then this menu will appear, and then you click on that, and then you click build list of dataset pairs. Okay, so therefore if you already have it, so this is the way to go. And when you click on it, so it will you will get this kind of an option. You can see it identified. There's a the forward and reverse minus _1 _2, and then it's going to combine them into one pair. So here, you need to disable this one, remove file extension, and if you want, you can keep the original elements visible. You can disable it as well. So here you can give a name, egdf, and then when you click the create list of pairs, so it will create our collection. Make sense?
Yes. Thank you, sir.
No problem. Okay, so shall I click on the create list of pairs? So then we can move forward. Others, you guys have any issues, let me know. Because this will be a learning experience for me as well, because I don't use Galaxy. I use the bash scripts to run run all these tools. But this is the one we all have access now. So therefore, Galaxies I only use for the educational purposes. And one day you will use either Galaxy or the more like coding style supercomputing or high performance computing approaches. Okay. Okay. So any issues? If there are, I assume there are no issues. So what I'm going to do is I'm going to create a list of pairs. Okay. So now you have a pair. Okay. So now we can move forward. Okay. So I hope all of us in the same page, like in the same step. So we can move forward. Let me bring back the presentation.
Sir, excuse me.
Yeah.
So can you explain again this? How can we get tool parameters?
Oh, you mean the FastQC format and other things?
No, sir.
Oh, the last one I did. So the last part I did, right?
No, sir. Not that one. So, come for this window, what should I do?
You mean this window, the window
sir.
I'm showing?
Input collection, tool parameters. How can we get tool parameters?
So I think you're asking about this, right? Here?
No, sir.
Okay. So
How can we
I will show you what the things I did and then let me know. Okay. So I think you're talking about this, right? This? This window, or what tool you're specifically focusing on?
Yes, sir. That's
This one?
Yes, sir.
Okay, no problem. Yeah. So you saw, right, what I did? I selected two files, and from the down arrow right here, I clicked on the down arrow, and then I selected build list of dataset pairs. And then when you click on that, you get this. And then what I did is, in my case, I disable both options, remove file extension and hide original element. And then I gave a name, because that way, I mean, it's easy for us if we keep them visible, because this is once you become expert, you can hide those things. But for the time being, so we need to keep our original elements visible. And file extension, if you remove it, sometimes some programs won't recognize it. So therefore, it's better you keep the file extensions. Okay.
Okay, sir. Thank you.
No problem. Okay, so we are moving forward, and I hope others don't have any issues. Okay, so the next thing we did is, I don't know if you want to see slides, I can show you slides as well. So let me, okay. So, and basically I asked to rename files, but that step, it's not really necessary. because you already learned about the things. And then this part is important. So please tag your the collection with the tag with this unfiltered tag. Okay. If you can remember, so you can just click on your data sets, and then you can tag. Okay. Here you can basically click at, and then I already typed it, so therefore it's already saved. So unfiltered, and then you save it, and then you go back. And I forgot to rename this one. MRSA save. Okay. Okay. So please add the unfiltered tag. Okay. And this is the procedure we followed. And if you can remember.
Excuse me, sir.
Yeah.
So can you do it again? Tagging.
The tagging. Okay, sure. It's very simple. You have your EGDF or whatever the name you gave. So here you can click on this icon. Okay. The pencil. Oh, sorry. Never mind. Sorry. This is a collection. So you click on it. Okay. And then you press this. Then the tags will appear. So here you need to click on this add tags. Okay. Let me repeat the process again. Give me 1 second. What? Oops. Okay, so my mouse pointer went missing. So you have your whatever the file collection, you click on it, and then you have this pencil icon here. Let me use the pencil should be okay. Okay, so here, so the pencil icon, and then you click on it, and then here you have add tags. When you click on it, so either you can type if your tag is already there, it will give you the whatever the available tag or tags. Okay, so then you click save. Okay, cool. Yeah, and nice. And yeah, what I was saying is, if you use a hash sign, so that means it will propagate into whatever the daughter activities you are doing. It's like the parent, and then whatever the offsprings it's getting. So basically, it will appear there. Okay. Okay. Let me go back to the presentation, and if then you can basically look at your the FASTQ files like last time. And then I asked 2 questions. So I'm not going to ask the same question again because sorry, I forgot one thing. So can you guys please rename your Zoom name to indicate either your last name, definitely your last name, or your index number. Either is okay. So hopefully there are not 2 Disanayakas or 2 Herats there. So therefore for a safe side, so you're whatever the name, please add your index number to the end. Keep the names, but on top of that add your index number, so that way I can trace back. Otherwise, because even last time, even after telling you, some of you use a username guest, so therefore I can't find who the guest is, right? So therefore the guest will get marks, but not the person who was representing the guest. Okay. Yeah. Okay, so please rename it. So and then to give you an answer for the first question. Remember the first one. It's basically some name to recognize the sequence and what sequence it used. So likewise, it can be kind of little long text. And then the second row we have the nucleotide sequence. And then third one, you have like, a plus. It's like, basically optional following a name. And then the last line or the last row, you have the quality scores because there we use the ASCII symbols. So each indicates a different level of quality. And I think we discussed about it like in a while back when we were doing our introductory lectures. Okay. So moving forward. Okay. And then we talked about the read quality improvement because when it comes to the sequencing, there will be errors introduced by like, the due to the different technical things also. So there can be errors based on like, whatever the quality of DNA or RNA we used. So therefore, we need to do some kind of a cleanup because we are asking this important question. So when it comes to our sequencing output, okay. So but like the what is the coverage? How much coverage our sequencing results have, and like how good the reads are, and then like do we need to like perform a new set of sequencing run rather than using our current data set, or like we whether we can salvage our sequencing if it's bad. Okay. So and also whether it will fit with our specific analysis, because sometimes if let's say we are studying the variations, we need a very high quality sequencing output compared to like the basic genome assembly, because if we are trying to like identify very specific variations, and if our sequencing results is messy or fussy, and then it is going to create problems. So therefore we are going to do the quality control. And if you can remember, last time we are using the tool called Falco. Falco is like more attractive mapper based on the FastQC, because FastQC is the most widely used sequence quality QC tool in the world, and it's basically developed by the Barham Institute. And so, but like there are some wrappers build on that fast QC in order to give a much better nicer appearance to like the fast QC output. So therefore, please go ahead and load the Falco tool. Okay, and there you can use the parameters, because remember, so it will be different from the last time. This is what we did last time. So now you have a data set, a data set of pairs. So therefore, accordingly, you can select both sequences at the same time. So go ahead and load both sequences to the Falco. Okay. Okay, and like if you face any issues, let me know. I will try my best to help. Okay, so I already close this, cancel, and go back. And we use the tool Falco. Okay, and then so you will see like you can see both, but like here. So we are using dataset collection. So you can not correct way, dataset collection. Okay, so then automatically your EGADF will appear. Okay, and then you can simply run the tool. Okay, clear? You guys have any issues?
So here we should select one file, one pair file.
Yes, because we have only two forward and reverse. Because I mean, it's beyond the 2 days activity. Sometimes we make a pair collection. So let's say you have like a sequencing run for like 10 different organisms. So then when we make a collection, we put everything together. So then under each accession number, we have forward reverse, forward reverse, forward reverse. So that's what we call as a dataset collection. But here we have dataset pair. So therefore, yes, the only one file will appear, but you will see when you run it, you will get two outputs. Okay.
Okay, so thank you.
Yeah. Okay. So others, I hope you're following the steps as we progress. So chat question. Okay, awesome. Hasnan, yeah, you got it correct. Perfect. Okay, so shall I run the tool? I hope all of you are already at this step. So I'm going to run the tool. Okay, so you can see the program is running. And there's like a small trick. So when you sometimes the galaxy.eu, as far as I remember, uh, unless you click on it, maybe it's coincidence, but I don't know. Sometimes it won't run until you interact with the data set. So therefore, you can try, because I every time I do it, when I click it, only the job started. Otherwise, job was on the queue and never ran. So therefore, just double check by clicking, I mean. Okay, so meanwhile, if you have any questions, so feel free to ask. Okay, so now it's running. And let's say if you let's assume you're doing a big project, but you can run it within a very short period of time, and assuming you're going to save all of your data. So because you can see we are given only 250 gigabytes, but if you click here, click here, you were given an option of using you can use 2 terabytes. Okay, but remember it's temporary, so therefore it will expire. So unless you save your output, one day when you log in, everything will be gone. Okay, so therefore depending on your requirement, so here this is a very small activity, but you'll see when you run in a big genome project, what is going to happen is, so your storage is going to get basically filled with data. So if you are going to experience that kind of a situation, so the ideal way is to run it in this scratch disk where you have 2 terabytes. And then what you can do is you can transfer your results into the regular 250 gigabytes space. And then you can move forward. Okay, so just a small tip. Okay, so we have our output. So what I'm going to do is I'm going to basically click on the web page, and then you can see we have results coming from both forward and reverse. Okay, so here it will give you like a very nice output as far as like well, I mean, what I want to say is like if you use the galaxy.org, you will see like a lot of gibberish. If you experience that, download the file. You can like click on the whatever the file, and then there is a download button. You can download it, and then when you double click it, it will open on your Chrome or like whatever the browser you're using. So then you can see the output. Okay, so if you can remember from last time, so whatever the things you in green it means good quality, and red means usually bad quality, but you need to double check them. And yellow means like medico, like in between medium quality output. Okay, and let me see. Okay. Yes, nice. Okay. Uh, And if you can remember, we this I mean this is a box and whisker type plot. You have like the median, so that is like the dark green one right here, and then the interquartile range, and also the 10%, 10% and 90% margins that indicated by the whiskers. Okay. Okay. So let's see. I hope everything is okay. Okay, so this slide is not needed. So next, if you can remember, so we are going to try to improve our quality. So that's where we ran into an issue last time. So let me move back into the slide. So what we are going to do is we are going to use the FastP tool. Okay, so now go ahead and load the FastP from your toolshed, and then see whether you see your data set. Okay, so go ahead first. And so then I will give you the options you need to pick. So here, like, now it's not like, the input one, input two, like last time. So here you can load your the pair collection. So go ahead and load it. And so you need to adjust to several parameters, including length of filtering option. So make it 30. And then read modification option. So pick yes for the cut by the quality in front of 5 prime and 3 prime. And also cutting window size, set it to 4. And cutting mean quality to 20. And under output options, keep the JSON file enable. Oh, yes. Okay, so these are the settings you need to follow. Okay, so I will keep the options on screen for a bit, and then I can show you it on the Galaxy environment as well. Okay. Okay, any issues? Okay, so shall I move into the Chrome window? So what I'm going to do, I'm going to go back and pick FastPay. And then, so here you have single end and paired collection. See, now we have the collection appears. It's unlike last time. Okay. And then under options, you have to pick several options if you just want look around. So there are some adaptive trimming options and global trimming. Okay. Overrepresented sequence analysis, filtering options. Okay. And then read modification options. There are so many options. So, but here, we are doing the very, very the kind of rudimentary or primitive stuff. So let me close all the options first. Okay. So, sorry. Yeah. And so here we are not going to merge forward and reverse, because sometimes if you do it we are basically combining both forward and reverse. So we don't need. Okay. And okay, so there's a question. Okay, Randika, you were saying a Galaxy user is stuck and not giving authorization even it activates and doing Falco in the Galaxy. Oh. Okay, so maybe you can try one thing. So refresh the screen. If it's not going to work, the other option is to basically sign out and sign back in. And if it asks for an authorization code or something like that, that's so probably it should come to your email and then you can enter. And also you can try cleaning up the cookies. Okay, so that sometimes work. So you have to try all those options, but it seems like your Falco is running. So therefore, see whether you can run all the options without an issue, without logging in or signing out. Okay. Okay. So keep me updated about your issue. Let's say what we can do. So meanwhile, so I'll show you how to put the different options. Okay. So okay, we filter options. Okay. And so the length filtering option. So right here, you can see under the length required. So we are going to make it 30. Okay. That's the only change we are doing. So you can read from the help what are the functions done by the other options. And then under read modification options, what we are going to select is, so there, here, the per read quality options. Okay, cut by 5' end, yes. Cut by the quality in tail, 3, yes. Cutting window size 4. And cutting mean quality, 20. This is the Fred value, remember. So Q20 means it's like 99.99% accurate. Okay, and then output options, you pick the output JSON report. Okay, so we already set the settings, and then we are going to run the tool. Okay, so I hope it's clear. So I'm going to run the tool. Hasnan, yes, good question. How we know the parameters? Okay, so when it comes to different sequencing runs, so people basically came up with the kind of a default settings after comparing many data sets. Yes, I'll show you the parameter slide. Give me 1 second. Okay, so these are the parameters. But depending on the requirements, let's say you run with default first, and then or whatever the protocol established by some person, and then see whether the quality improves. If not, we are going to modify them in order to obtain the desired good quality sequences. Okay, so there's a big help manual if you're interested. Every tool you load, so there is in the bottom, right after the email notification. So there is kind of an area talking about the quick help and also the link to the major tool. So you can go to the tool site and see what are these parameters and how we can play around. Okay, so each heading will be described. Uh, I mean, it is already described in the user manual. Okay, to just talk about these parameters, like so remember when sequencing happened, so 5' end and then the 3' end, there will be fuzzy areas, even if you think about Sanger sequencing. So therefore it's better to cut the from front and from the back if the sequence quality is not good. So I mean it's like a simple translation of these sequences. And also some tools, so they basically go by the slides, frames. So here we are using a cutting window of 4. Okay, so when it cuts for and then when they cut, so the program will look for Q20 less than Q20. So that means the poor quality sequences. So likewise, so therefore, you can pick parameters based on there should be a fundamental reason behind it when you select your parameters. Okay, so I hope it's clear. So I hope you grab the parameters. Sorry, excuse me. Okay, so shall I run the tool? And Hassan, I hope it's clear. So I'm going to run the tool. Okay, so now our data set is so basically going through this the quality improvement process. If you guys have any questions regarding this step, so please feel free to ask. And the good thing with the galaxy.eu is it will show you different comics, and so you can have some fun while working with your data. Okay, so very good. Yeah, awesome. Okay, so that was fast. So we have our output. Okay, so now, so our data is improved, right? So therefore, we need to change our tag. Okay, so here we have the unfiltered, right? So now we need to change it. How you do it? You click on sorry, I always do that. So let me go back. Okay, so you basically click on the title of the file, and then you can click this one, and then you can remove. So now we will add a new tag, okay, called filtered, and then you can save it. Okay, so you can do the same for the other files as well. And filtered. Okay, save. Go back. Sorry. Oh, sorry. Yeah. See here. And do the same for this. Filtered. Save. Go back. Right. So those are the outputs from FastPay. Okay, very good. Yep. Okay, so go ahead and change the tags. Okay, so so now we are going to look at the HTML report. Okay, so yeah, go to the fast. Let me go back to the slide. Okay, go to the FASTP HTML report. Okay, and see what are the things given there. Okay. So and then you should be able to answer these questions. Okay, so let me. So go ahead and answer the question by looking at your data. Okay, so there are 4 questions. Okay, so the first question is, how did the average length change before and after filtering? So you can just say, okay, so before filtering, it's how many base pair, the range. Hasnat, the reason for the tags disappearance is basically you need to click the save button. Otherwise, they will disappear. Make sure you it's there, and then you save it, and then you go back, and also check the top. Sometimes you will see somehow it the tag unfiltered is selected, so you need to basically delete that from this the tiny search bar on top. So that may be the reason why the tags disappear, because the unfiltered were filtered out from filtered. Okay. Cool. Okay. So remember, these questions will carry marks. So therefore, if you answer, you get marks. If not, no extra credit. Simple as that. Okay, so we will have a 5 minutes break. I will keep the questions open, and then Dika, you want to see the FASTP process? Okay. Others can take a break. I'm going to show Randika the process. Okay. So once you have the Falco output, not Falcon, Falco output, what you do is you basically click on the FASTP.
Yes, I did.
Oh, you did that?
No, sir, I opened, but not the sequence. I mean that process, I didn't follow.
Okay. So you basically go select the FASTP, and then from single endo pair and reads, you need to select paired collection. Okay. And then so whatever your output will appear there. because your case, it should be this, the unfiltered one. And then I will display the parameters. So you can basically set those parameters and then run the tool. Okay, so let me show you the parameters. So yeah, so these are the parameters you need to put, but this part you already picked. So the focus on the filter option, read modification options, and output options. Accordingly, you can modify the settings. Okay.
Okay, sir.
Yeah.
Thank you, sir.
Asnad, you are looking at the JSON file. You need to look at the HTML file because HTML file will have like a very nice output. JSON file is for a different purpose. You don't need that to answer this question. So go ahead and check your HTML file. Okay. Any issues? I see like, four of you, sorry, six of you got, answered the quiz. Okay, so eight. Awesome. Okay, so I will keep it open for maybe another 30 seconds. Meanwhile, any other questions? Okay, so if you see like, like some black file with like, some codes, so that's the JSON file. What you need to look at is the HTML file. Okay, it's more human readable than the JSON file. Okay, so I'm going to close the quiz. Let me end the poll. And go back, close. Okay. So moving forward, we now have the FastQL HTML report. And if I'm pretty sure you guys noticed that before filtering, it was like, between 190 base pairs to 221. And after filtering, it went from like, it kind of like it went from 190 to 189. And then the other end, 219. And it also increased the Q20 and Q30 bases. So that means like, there's somewhat quality improvement. And third question, trimming affect the GSC content. There's no significant change and it's expected. And the data, it's not the awesome data, but definitely we can use it to move forward. Okay. Madhushika, let me see whether I can get the question back on. Give me 1 second. Okay, so Madhush. Aspi. Launch. Okay, so I'm opening the poll for like in a few seconds. And meanwhile, this question from Minoka. Okay, so you're talking about the JSON file, right? HTML report. So this one, like, you're asking about the tagging. So it's same way. You click on the file name, and then here from pencil icon, you can basically select your tag because if you click it, since the filter tag is already created, So then you can basically select it, because here right now I already selected it. So therefore I have only the removal option than the adding option. So let me remove it, and then I can basically add it, and then you click outside, and then you click save. And regarding the JSON file, the second point, I mean, what I mentioned earlier was like, so it's not necessarily a human readable. It looks like this. And even if you click pretty print, a print for printing is fine, but still it's not so pretty to read. And it's not human readable. So, and what we are looking at is basically the HTML file. So where you have your output. Okay. Okay. So we have before filtering, after filtering, and then the filtering results, and you can see whether it detected adapters and all the the information. Okay. So before filtering, and then okay. So you can see base content and so many parameters. Don't get overwhelmed by this output. So eventually you will learn the importance of these different parameters. But here today we are more focusing on just the very very simple things related to our data. Okay. Okay. So where were we? Okay. So I'm going to close the poll. Okay. And I hope Madhishika, you already added your stuff. Let me close it. And then we are going to... Oh, okay. So Hasna, your question is what is the purpose of JSON file? When you do the molecular biology, this computing stuff, JSON is kind of the format we use to feed the other programs. And so this file is very comprehensive. It contains all the information, settings and everything in a single file. So that is very good for the documentation process as well, but it's not necessarily human readable. So therefore, most of the time, we will use it to archive everything we did rather than the visualization things. Okay. Uh, so now we have our improved data. So next, since remember I told you we are talking about the MRSA, the Staphylococcus aureus, and now we need to see whether our data brought our species back into whether we have our expected species there. Okay. So, and the question is why we are doing taxonomic profiles. So in this case, as I mentioned earlier, we want to see whether it's a Staphylococcus aureus and what kind of a strain of Staphylococcus aureus. Likewise, so that way we can make sure that because this is more like diagnostic setting as well. So therefore you can say, okay, so this species is, or this strain is there in this the sample or the patient sample, and whether it's a new species or an existing species, or it's there's a lot of mutation although it's the same strain. So those things we can basically identify from the data. Okay, so what we do is we need some tools to do that. So therefore, we have to have some kind of a reference database, and based on that, we can classify our samples. Okay, so to do that, we use one of the most popular tool, the tool called Kraken. Okay, so I am pretty sure if you watch movies, you heard about the Kraken, that thing comes to destroy the world. So you can check it afterwards after the class. Okay, so the idea is here what we do is we have our the reads, and then we are going to basically map into a taxonomic tree. Okay, so based on a pre-built database. So in very very simple term, what we do is we have let's say 10 species, and then we have our reads, and these 10 species we know all the taxonomic levels, and then we put basically feed our reads and see what kind of a taxon or the taxonomic hierarchy they are going to match. You will see the results in a bit. It's what we call as a compact hash table, and they use in technical terms probabilistic data structure, and so that help us to do queries very fast. Otherwise, it will take some time. This is a very optimized tool that you can use in order to analyze your outputs, especially if you do metagenomic studies. This is one of the go-to tool people use in order to identify, and that way you can map to the whatever the remember the common ancestors based on the species tree that you can do with this. And so it's a pretty cool tool. Okay, so and then oh sorry, my bad. I forgot to project the slides. You guys didn't mention that. Okay, so this is how you do Kraken. Okay, so remember I told you there's a query sequence, and then we match it to a pre-built taxonomic tree, and then we will see how they hit into different ancestors, and then accordingly we are going to build the taxonomic classification of the different taxons these trees belongs to. That way we can go up to even strain level with this tool. And if I, as I told you earlier, it's a very, very efficient, less energy consuming tool people use, especially for the metagenomic assays as well. Okay, so what you're going to do is, so go ahead and go to your tool shed and select Kraken 2, because the earlier version was Kraken and now it's even more further improved. So now it's called Kraken 2. So go ahead and click type Kraken 2 on your tool shed, and then from there you can select your data, and there you can set these settings. So set the minimum base quality to 10, and under create report, select yes for the print report with aggregated counts and clades to the file. And as the source database, click plus PF16. Okay, so just double check. Sometimes that database not available. If it's not available, I'll give you an alternative option. But so go ahead and load the Kraken tool, and then set these settings. Okay. So for the sequences, you're going to select the output came from the FASP. Okay, so not the original EGADF data pair. Okay, so go ahead and do it.
Excuse me, sir.
Yeah.
So here I have plus PF 21 only.
So the plus PF 16 option is not there, right?
No, sir.
Yeah, so that's okay. So you can select that. So let's see what kind of output we are going to get. No problem.
So
don't worry about it. And if you check the database.
Thank you.
Yeah.
So I think I saw the option.
Oh, so you see the option, right? So
Yes, sir.
yeah.
Plus
So
16, yeah.
Yeah, so I mean that's the thing. Sometimes even when you're in the same Galaxy interface, there are different versions. So therefore, you see different the databases available. So, but you can improvise, because if you look at there's a latest database which is little bigger. So that's why I didn't suggest, because it will take more time because we have a limited time. So we are going to go with this one. But so select whatever the closest option. We'll see what kind of output we are going to get. No problem.
So, excuse me, sir.
Yeah.
So, it is paired one, right? It is not paired collection. In first step, there is single or paired reads. That is paired.
So there, we can check.
I have paired collection.
So let me go there and then Kraken 2. Okay, so there. So here, you need to select the paired collection, because we did our FASTP on the collection, right? So therefore, so basically you need to select the FASTP on collection 5 paired end output, right? This is
Okay, sir.
makes sense?
Excuse me, sir.
Yeah.
So here appear as minimum hit groups as 2. Is it correct or should I
Minimum hit groups.
hit groups? Yes.
2 is okay. I mean, that's the default. We can change it, but like you need to change the minimum base quality to 10.
Okay, sir.
Yeah.
Okay, thank you.
Yeah, others, you are facing any issues?
Excuse me, sir.
Yeah.
When we consider the GC mean changes, so what is the minimum changes to when we consider as the significant changes?
That's a very good question. So let me go back to the file. So let me quickly run this one. I forgot to run it. Then I will go to the file. Give me 1 second, that's not. Because this minimum base quality 10, create a report. There print select recommend rebuild plus PF16 yeah plus 15 16, and let me run the tool. Okay, so your question is on the FASTP. HTML file. You're talking about this, right? The minimum GC content.
Yes, sir.
So this is like kind of an approximation because the before it's 32.2.24 and then afterwards it's 32.19. So I mean, so you're not going to do like a statistical analysis because if you think about like so it goes from 0 to 100 now. So like in that for particular species, it's around 32. And then like the main value even like up to like the decimals, so that's the place it changes, so therefore it's like approximation. So no, there's no statistical test done. It's like just you eyeball and then say it, because if you look at the distribution, like you will see that it will be a curve. And then if you look at the second one, so it will overlap because we allow some flexibility within that, like when it comes to the distribution. So therefore, it's negligible. But you can't do like a statistical test. I mean, if you want, you can do. I'm pretty sure it will come non-significant as well.
So normally we are analyzing mean using the mean separating method. So in that case, you asking there's a significant difference or not. So that's why I asked. So how can we? Yes or no? We have to clear no. That's why.
Yeah. I mean, like I get your point. Like like I didn't expect to go like that deep. So like because it's not even changing by 1%. So therefore, I mean, based on that, it's an approximation. So, but don't worry about it. Like about like mean separation and like no, don't think that hardcore. I mean, so this is like a very simple exercise. So, but don't worry, there will be no negative marks. Okay. So let me go back. Okay, so the Kraken is going to run for a little bit. sometimes takes like 5-6 minutes. So meanwhile, I would like to show you a video. Okay. So because here, like, remember I talked about the metagenomics, and metagenomics is like one of the major fields when it comes to exploring genomes. So here, like, we are looking at multiple. So let's look at this video while our program is running. Okay. I think I need to share it again because I forgot to select sound. Let me quickly change the settings and share sound, desktop to, okay. All right. So let's watch the video. Let me mute myself.
Hello, welcome to watch the CD Genomics' video about metagenomics. Let's begin with what metagenomics is. Metagenomics, also known as environmental and community genomics, is the genomic analysis of microbes by direct extraction and cloning of DNA from a microbial community. Metagenomics provides a culture-independent insight of microbial communities. It has emerged as a powerful centerpiece, since most microorganisms cannot grow in a pure culture, and that culturing cannot capture the full spectrum of microbial diversity. The obtained community taxonomic profile can provide massive amounts of information on genomic assembly, gene prediction, species diversity. Comparative metagenomics can provide additional insight into the function of complex microbial communities and their role in host health. Therefore, metagenomics has the potential to advance knowledge and solve practical challenges in a wide spectrum of fields, such as infectious medicine, engineering, agriculture, sustainability, and ecology. Metagenomics approaches are roughly classified into two groups: whole metagenomics and targeted metagenomics. Although whole metagenomic analyses revealed that microbial communities are well adapted to their geochemical conditions, they can't provide definitive evidence for the positive selection of enzymes for key ecological processes under environmental pressures, likely due to insufficient sequence data for the target enzyme group. Targeted metagenomics is a suitable tool for constructing gene collections of specific groups of enzymes which are useful for studying their adaptive evolution. Whole genome shotgun sequencing, WGS, has been increasingly recognized as the most comprehensive and robust approach for metagenomics. It offers the advantages of identification of species-level taxonomy, and the estimation of metabolic pathway activities from human and environmental samples. Several large-scale metagenomic projects have been recently conducted, or are currently underway utilizing WGS. With the generation of vast amounts of data, the bioinformatics and computational analysis of WGS results become vital for the success of a metagenomics study. However, WGS has several limitations. First, each step in the WGS data analysis, including metagenome assembly, gene prediction, taxonomy identification, function annotation, and pathway analysis, is complicated by the sheer amount of data. But algorithms and tools have been developed specifically to handle WGS-generated metagenomics data, with the hope of reducing the requirement on computational time and storage space. Second, one of the biggest considerations for library preparation of environmental samples for shotgun metagenomic sequencing has to do with amplification. Certain types of samples, such as water and swabs, yield small amounts of DNA, necessitating amplification during library preparation. Amplification by PCR can over-amplify certain fragments over others. If you're able to extract enough DNA material, about 250 to 500 nanogram, an amplification-free based library preparation method is recommended. Targeted metagenomic analysis involves isolating DNA from an environmental sample, cloning the DNA into a suitable vector, transforming the clones into a host bacterium, and screening the resulting transformants. The clones can be screened for phylogenetic markers, such as 16S ribosomal RNA, or for other conserved genes by hybridization or multiplex PCR, or for expression of specific traits, such as enzyme activity or antibiotic production. High numbers of novel biocatalysts have been identified by function-based or sequence-based screening of metagenomic libraries derived from various environments. The sequence-based approach has been used extensively to retrieve specific genes, from a pool of DNA. Instead of cloning all the extracted DNA, primers are designed specifically against an identified target gene. The advantage of using sequence-driven screening is that it uses well-established and high-throughput techniques, such as PCR and hybridization, and can be used for different targets. For example, the 16S ribosomal RNA gene is a taxonomic genomic marker that is common to almost all bacteria and archaea. The portrait of the microbial world was revolutionized by analysis of 16S ribosomal RNA genes. Nevertheless, when it comes to fungi, there is a different scenario. Instead of 16S, 18S is commonly used in fungi for phylogenetics, since it has more hypervariable domains than 16S. In addition to this, the internal transcribed spacer region, also known as ITS, removed in the post-transcriptional process of nuclear ranitchistrin, has been widely regarded as a universal fungi barcode marker for a successful identification for the broadest range of fungi. And compared to 18S, ITS is more variable, and hence more suitable as the genetic marker for measuring intraspecific genetic diversity. Functional metagenomics studies the functions of encoded proteins. Functional metagenomics involves isolating DNA from microbial communities, cloning DNA fragments, expressing genes in a surrogate host, screening for enzymatic activities, sequencing, and functional analysis. When it comes to the construction of a metagenomic library, COSMID or FOSMID-based libraries are often preferred, due to their large and consistent insert size and high cloning efficiency. DNA is first extracted from the environmental sample of interest, then size selected, and repaired, and ligated to a COS-based vector, allowing packaging by lambda-phage for subsequent transduction of Escherichia coli. The resulting library contains relatively large insert DNA, typically 25 to 40 kb for COS-based vectors. Functional analysis has identified novel antibiotics and antibiotic resistance genes. Thanks for your attention. For more information, please visit our website www.cd-genomic.com. We also provide comprehensive metagenomics services to meet your specific need. We are more than happy to be assistance. Hello.
Okay. So there you go. So how many of you got your output? I did. What about you guys? Have you obtained your output?
Not
Okay.
yet, sir. Still running.
Okay. So how about others? You can type yes or no. So then can move forward. And meanwhile, okay, so still running, right? Yeah. Okay, Hasnat, you got it. And Randika, so have you started Kraken 2?
Yes, sir. Got the result. It is
Okay.
about 6-column table.
Okay. Okay, sounds good. Yeah. So we will wait a little bit because for some of them, I mean, like some of your colleagues, so they're still, I mean, the program is still running. And we'll wait like probably like, a couple of minutes and then see the output. So you should get like two files, classification file and then the report file. We are going to start with the classification file. So go ahead and those who got results, check the classification file and see whether you can interpret what is given there. Mm. Yes, a bunch of numbers, right? So see whether you can find what is the meaning behind these numbers. So others, can we move forward? Those who are still waiting for the results. Did you get your result? Okay, so Hasnat's question, how much these results are, or how precise the results are. Okay, so it's based on the sequence similarity. Like remember, you did BLAST. So likewise, what we are doing is we have our read sequences, and then we use the IMers came us earlier, and then we basically match it. And when you think about the accuracy, we can go up to strain level accuracy. So that means a very precise, as long as we have the sequences the of whatever the strains inside our database, because we are not necessarily try to find new species, but rather we are going to match the species with the pre-built database. Okay, so and then let's say we have new things, unmatched one. So then we can further. I mean, not use this tool, but there are other tools that we can use in order to further classify or further taxonomic identification of those strains or species. Okay, so shall we move forward? Okay, so let me show you the PowerPoint file. Okay, so the output. So it's a tabular file. Okay, and so you can see each sequence is basically classified by classified by the program, the Kraken 2 we use. And if you see if you look at the first one, it says C or U. C means classified, U is unclassified. So likewise, so these are the meanings. And the second column is showing the sequence ID, and then the next column is giving the NCBI taxonomic ID. I'm pretty sure you guys learn about the NCBI taxonomic database. Okay, so here, based on our sequence similarity to the pre-built database, it will tell you what kind of a NCBI taxonomy ID it was assigned. Okay, and the 0 means it's unclassified, and then it will give you the length of your sequence in base pairs, and the whatever this I forgot the name of that sign, the separator. So there, for read 1 and read 2, I will show you an example files in a bit. And then there will be a space delimited text indicating the lowest common ancestor mapping for each KMA in the sequence. Okay, and to give you an example, if it says 560 to 13, 561 4, A31, likewise, it indicates that the first 13 KMAs were mapped to the taxonomic ID 562. Okay, so the next 4 must mapped to the taxonomic ID 561. Okay, and then the 31 means because it's the the ambiguous ones. Okay, so the remaining 31 must map to the ambiguous, and then the next the fourth line indicates those KMAs were not in the database we provided, and then it's followed by the last 3 KMAs, and those ones were mapped to taxonomic ID 562. Okay, so that's what it indicates. And this indicates the sign, the end of the first read, and then this start of the second read, like the separation. And so I'm pretty sure, when you look at your output, it is like this. You can now match your columns with the columns mentioned here, so that way you can get an idea. Okay. Okay, so this is basically an overview. So then I will ask a couple of questions regarding your output. Okay, so let me know if it deviates. I mean, not necessarily this, but when you answer the questions, if you see any deviations, let me know because for some of us we had to use a different database. Okay. Shall I move into the questions?
So it's still running, so.
Yeah. Okay, so I mean, don't worry too much about it. So let it run. So probably at the end, I will give you a link. So then you all can do it at your convenience. And Randika, so you got different data. So what is the database you use? What is the pre-built database you used? Was it the PF18 or a different one? Can you share a screenshot in the chat window? Okay, so when the for the NCBI taxonomic ID, you can simply search the taxonomic ID inside the NCBI database, NCBI taxonomy database. So then it will direct you to the relevant species. Let's say if you type 561 and search, you will find the corresponding organism. Okay, so let me ask a couple of questions regarding the report you got. Okay. And go to pause, quizzes, back, track and classification. Yeah, so go ahead and those who got the results, so you can answer the simple quiz. Okay, so regarding the second column, so it may vary. So yeah. So we'll see. If it deviates from whatever your result, so you can use the chat box and type the taxonomic ID you got. 1 and 3, definitely you can answer. Second one, most probably 99%, I guess. Not like I'm not doing any mean separation like Hasnan mentioned to us earlier. Yeah.
Excuse me, sir.
Yeah.
So that in the two outputs, so one is the classification and another thing is the report only. So
You're looking at the report file.
yeah, so I need to look onto the report file or the classification.
The report file.
Report file. Okay, sir.
Yeah. Mm-hmm. Randika, you are looking at the wrong file. You are looking at the other file. You need to look at the report file. There are two files you get as your output, but you are looking at the wrong one. The other file contains like a lot of numbers, like the example I showed you. Okay, only one person answered so far. Others?
Excuse me, sir.
Yeah.
Sir, I have a question. For the Kraken tool, what kind of file should be used?
So you mean like, so he has an input.
Yes, sir.
So here, so you go and click paired collection.
Yes, sir. I selected that.
And then it will the first option you're getting is FastP on collection 5. Like in my case, it can be different number, paired and output, because you will get like a two options. If you
Yes,
follow
sir.
the same procedure, what you need to select is the filtered version.
Okay. Sir.
Yeah.
I selected it and I ran, but still running. Sir.
Okay, so I mean that's okay. Yeah. So what we are going to do is so we will I will be we will wait probably like another 5 minutes for you to get the output, and then I will share the link to the tutorial with you all, and then you can follow the whole process. Okay, so this is adapted from already available Galaxy tutorial. So therefore, there's a very nice video presentation, and also step by step procedure, so that you can follow at your own pace, and then please go from the beginning to the end of the tutorial, because starting from next week we are going to assemble uh, a genome. Okay, so this is a very very crude version of thing that we did. We were more identifying, but what we are going to do is, as the last part of your course, we are going to assemble a genome, and you will be getting your own customized genome. Okay, so therefore you need to be very familiar with the whole process. So therefore, practice it. So that way we can have a very nice smooth experience when it comes to assembling our your favorite genome. Okay. And I hope last week, this week, and a couple of weeks before. So we worked on the Galaxy platform. So therefore, I hope now we are more familiar with the interface. Someone was trying to ask questions. So please go ahead.
So what is about the third question? I don't understand. Taxonomy. Is it from the same file or from others?
No, because if you can remember, so whatever the number you're getting correspond to a species, right? So what you do is basically you type the number you're getting, and then you search it inside the NCBI taxonomy database. So then it will tell you what kind of a species it is. Got it? Whatever this the number starting from, let's say 1279. So that is the taxonomic ID. So what you need to do is you need to find what species correspond to that specific taxon. Got it?
Yes, sir. Thank you, sir.
Yeah. And so far, most of the questions, only 3 or 4 people, provided their answers. So, I mean, say the coming the genome assembly activity, so there is a portion of marks coming from towards your final exam, not like extra credit but as real credit. So therefore so your active participation and the following the procedure is very important. So just I just want you all to know about it because so I want all of you to when you finish this course, at least you should be able to read some protocol, and then you should be able to come up with your own solution. So that is the whole idea behind this course. So therefore I want all of you to be in the same page when it comes to activities like this, and handling data and analyzing data, etc., etc. Okay. So therefore, please think about that as well. And I'm going to stop at this point today. But I mean, I will keep the quiz open for another 5 minutes or so, so you can answer the quiz, and then I will email you the link to the tutorial, so you can follow it at your own speed, and then please try it, and then get yourself familiarized. Okay. So those who already answered the questions, so if you don't have any questions, you can leave. But if you have question, you can stay, and the others. So the quiz window is open until probably another 3-4 minutes. So you can provide your answers. Okay. So have a great evening, and I will see you guys next week. Thank you. So that's a very good question, Hasnan. Depends on our requirement. So let's say if I work with let's say rice plants as an example. So then we are focusing on one individual. And but when it comes to metagenomic sequencing, we are interested in more than one species. We are looking at the whole population, and based on the reads, we can basically identify multiple species. And with sometimes to give you an example, I was working with potato genomes, and so there I use Kraken tool to identify whatever the other viruses present in that same sample. So I mean that we can with the help of these kind of tools, we can go beyond the scope of our studies, but there are different processing steps in only in the sequencing, especially when we make libraries, we have different targets. Depending on that, it will decide whether we are going to look at multiple species or single species. So therefore, it varies. Yes, of course, we can look for multiple species at once. That's what people do when they do metagenomics. But if you're specifically focusing on a genome, so then it is a different process you follow. So depending on the process you follow, you can either go towards more species or one species.
Excuse me, sir.
Yeah.
It's but also when we considering the two same species but different individuals.
It's like population genomics. Yes.
Yeah. But when we considering now we going to the sequencing project. So if there are more than one individual, if available, we can get the bunch of all the genome sequencing details. So how can we divide the particular this report for this individual and this report for this individual? That's what the question. That was the question.
Okay. Yeah. So there also we use this the cool thing. What we do is when we sequence, we put an index tag. Okay. So when we prepare the library, so let's say you have sample coming from individual two, we put different tags. So that way, so when we sequence them collectively, but they have a tag. So therefore, based on the tag, we can divide. Okay, so this is the individual one, this is individual two, this is the individual three, likewise. So it can be easily done. I hope I answered your question. Okay, so if there are no other questions, I am going to close the poll. And then, as I promised, I will share the link to the tutorial, so you all can so practice it at home. Okay.